Welcome to our first topic in chapter 10, which is genetics and evolution. We are going to do a pretty deep dive into meiosis. AHL stands for advanced higher level. You have covered, in a general sense, meiosis in freshman biology, and you also covered it in year 1 of IB Bio, but now we're just going to go over it again, make sure you guys have all the bits that you need to be successful for this chapter. So let's just talk about what meiosis is. It is a reduction division from a diploid somatic cell, 2N, to a haploid gamete. The chromosome number is reducing, right? Going from diploid to haploid. You cannot have a diploid gamete because when your gametes come together, you need to be able to add them to make a viable offspring. So if you add 2 diploid cells together, you have too many chromosomes, whereas if you add 2 haploid gametes together, you get 1 diploid cell. So let's start from the beginning. There are 3 phases, G1, S, and G2. G1 is where cells grow and synthesize protein. S is like ding, 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 star. Make sure you know that the S phase is where DNA replicates. And then G2 is another growth period. This is kind of where the final checkpoint is before the cells start going through a meiosis. So once the chromosomes copy during the S phase, they attach at the centromere and are now known as sister chromatids. So in prophase 1, you have homologous chromosomes that have joined together to form what's called a bivalent. So this kind of set of 2, right? You have like this orangey one and this purple one that has formed a bivalent. Crossing over between non-sister chromatids can take place here, so you could get like the closest orange one cross over with the closest purple one. So the ones that are closest to each other would cross over. You would never have like this first one and this fourth one cross over. They're just too far apart. And this is a great source of genetic variation in gametes. So let's talk about crossing over for just a minute. So there's kind of like 3 stages. The synapsis is when homologous chromosomes associate with each other. That's when they like come together. And then the chiasma formation is when the chromosomes actually cut at the same spot and form what's called a holiday junction. And the holiday junction is where the DNA is swapping. So the DNA of the cut sections will attach to the opposite chromatid, and then you end up with what's called recombination, which is the alleles that now have non-sister chromatid ends on them. So this idea of swapping genes in this crossing over is way more likely to occur between genes that are further away from each other. If the genes are really close to each other, it's unlikely that the genes are going to be able to like cut nicely in between them, right? That chiasma is not going to be able to cut in between them, but if they're further away, it gives it a little bit more leeway to separate the genes. After prophase 1, you go to metaphase 1, where the bivalents line up at the equator and random orientation occurs. Random orientation is the idea of the homologous chromosomes just lining up in any random order. And you can see that in the bottom. I have this pink one, and then blue, and then orange, and then purple. But in this next one, the orange and purple have swapped places. In the next one, the blue and pink have swapped places. So it doesn't matter how these bivalents line up at the equator, but how they do is totally random, and that's the definition of random orientation. And then anaphase 1. So this is where the spindle fibers contract on those centromeres and pull the homologous pairs to the opposite poles. They're separated. This is the reduction division. Highlight this, star this, underline this. This is the reduction division. And this is where non-disjunction can occur and can affect the chromosome number of all 4 gametes. So if the homologous pairs don't separate properly, you can have non-disjunction, and it will influence all 4 of the gametes that could be produced. After that, it's telophase 1, where you have new nuclei form and cytokinesis begins to take place. The nuclei are no longer diploid, so now we have haploid nuclei. And if crossing over and recombination has occurred, then we do not have exact copies of the sister chromatids anymore. So now we have some genetic variation in our cells. After that, we go back through again, because meiosis has 2 rounds of this cell division. So interphase happens again, but there's no new synthesis, and it sort of like happens, but it's really quick, and you don't spend a lot of time in it like you would in the initial interphase. In prophase 2, you have the breakdown of the nuclei, and no additional crossing over occurs. Then you go to metaphase 2 and anaphase 2. Again, you have the same general idea happening. You have the sister chromatids aligning at the equator. Random orientation can happen, but you don't really get, it doesn't really contribute to variation as much as in metaphase 1. So while, yes, it's important, it's not as important as in metaphase 1. And then anaphase 2, same thing happens. Spindle fibers contract, the sister chromatids are pulled to opposite poles, and non-disjunction here will lead you to 2 of the 4 gametes with incorrect chromosome numbers, whereas if you had non-disjunction in anaphase 1, all 4 of your gametes would have incorrect chromosome numbers. Now, I mentioned that there's some genetic variation, but not as much, and so metaphase 1 verse 2, you have more in metaphase 1 because it's random orientation of homologous pairs, so there's a lot of possible combinations, whereas in metaphase 2, it's random orientation of sister chromatids where there's really only variation wherever crossing over occurred. So if crossing over didn't occur, then there's no variation happening. Then we have telophase 2, where you have your haploid nuclei formed, cytokinesis begins, and the end result is 4 haploid gamete cells. So that is the end result that we're looking for, and that's what we care about. Now again, genetic variation, that's like lights flashing, the main thing we care about in meiosis, right? Genetic variation that leads to a successful population. So there's 3 genetic variation causes in meiosis, crossing over in prophase 1, and then random orientation in metaphase 1 and 2. We've talked about those 3 already. The last one is random fertilization and sexual reproduction. This is the idea of 2 different people coming together and reproducing. So getting gametes from a mom and a dad that are 2 different people leads to genetic variation as well. So now we're going to dive into our favorite person. He's not my favorite person, but he's a person that I know and studied very well, Gregor Mendel. So he came up with a bunch of laws, and if you're unfamiliar with the laws, please go back to chapter 3 and check out some of those videos on chapter 3 so that you can go down memory lane with good old Gregor Mendel. So one of the laws that he came up with is this law of independent assortment. And what this says is that the presence of an allele has no influence over another allele. So basically what this means is just because you would inherit, a capital A, looking at these genes over here, just because you would inherit a capital A does not guarantee that you would inherit a capital B. As you can see, you could get that, but you could also get a lowercase b because there's no influence from one gene to another. Of course, this only holds true for unlinked genes. If these genes were linked, then yes, they would be inherited together, but that's not what this is talking about because Mendel is the father of classical genetics. So this is assuming that everything goes perfectly according to plan. So independent assortment, again, is this idea that one gene is not guaranteed to be inherited with a second gene. So just because you have brown eyes does not guarantee that you have brown hair, right? You could have brown eyes and blonde hair, brown eyes and red hair, because they are not inherited together. So a lot of people get these 2 things mixed up, random orientation and independent assortment. So random orientation is the behavior of homologous pairs in meiosis, right? So it's how the homologous pairs line up in metaphase in meiosis. And independent assortment is the behavior of the alleles of unlinked genes as a result of meiosis. Now I know that this isn't like super different, so this last sentence is like kind of important. So due to random orientation of the chromosomes in metaphase 1, the alleles of these unlinked genes have become independently assorted into the gametes. So Mendel, way back in the day, understood that independent assortment happened. He didn't really know why, because he couldn't figure, he like didn't have the tools to be able to see how these homologous pairs of chromosomes were lining up in metaphase, but he knew that they were assorting independently of one another. And then random orientation is the reason why that they assort independently. But it is two different things, and you need to understand the difference between the two. So that's all for today. We'll go over this a little bit more in class. Nice job.